Thank you very much for the invitation for the International Congress of Photosynthesis Research in New Zealand. And I'm going to talk today about the network and systemic acquired acclimation and cellular light memory in plants. I start from the introduction and just a couple of basic information. Yes, when the light is absorbed by photosystems, it is immediately splitted on the three energy channels: fluorescence, photochemistry, and heat. And plants can regulate this energy flow between these channels. In normal condition, in the field condition, photochemistry is saturated at the relatively low level of light intensity at between 400 and 600 microeinsteins. And then we can increase the light. However, the photochemistry and photosynthesis is not going to be increased. Energy still is absorbed, so it is absorbed in excess, what can be utilized by the photochemistry. And this energy is dissipated or quenched by different processes like water-water cycles, non-photochemical quenching and regulation of photosystem 2, photorespiration, and recently discovered, 10, 12 years ago, quantum light memory, cellular light memory. Just to for indication and to remind that non-photochemical quenching is described by the mathematical formula and it can be defined as a ratio of the maximum fluorescence to the maximum fluorescence prime minus 1. And this is quite important because when we know that NPQ is described as by mathematical formula, but it is described on the basis of modulated fluorescence parameters. It should reflect the heat channel. However, I will show in the presentation that it is mostly reflecting the fluorescence channel. And in 2004, it was presented by Pick et al. In PNAS, a very important paper that evidence for a complex collective dynamics and emergent distributed computation in plants. In that time, for the first time, it was shown that images of the fluorescence are not equal on the whole leaf surface, and they have a spatial pattern with regions which are higher fluorescence or lower fluorescence. And they calculated that in fact these fluorescence changes in the cells are somehow connected with the stomatal conductance, and whenever the fluorescence is higher, the stomatal conductance is lower. Whenever the fluorescence is lower, then the stomatal conductance is higher. This was a quite important finding. Now I would like to talk about the discovery of chloroplast retrosignaling for nuclear encoded genes. Ascorbate peroxidase is dependent on redox status of the plastoquinone pool and ROS signaling. In 1997, we published the first paper showing that the photosynthetic electron transport regulates the expression of cytosolic ascorbate peroxidase genes in Arabidopsis during excess light stress. In this paper, we shows that induction of ascorbate peroxidase 1 and ascorbate peroxidase 2 is strictly dependent on the redox status of the plastoquinone pool and on the chloroplast redox signaling. Later on, 2 years later, we discovered systemic signaling and acclimation in response to excess excitation energy in Arabidopsis. And in this paper, we found that local and systemic induction of ascorbate peroxidase 2 is depending on hydrogen peroxide signaling, is depending on the retrograde signaling from the chloroplast, depending on the redox status of the plastoquinone pool. Later on, we added to this signaling abscisic acid and other components like electrical signaling, and generally we know that the cocktail of reactive oxygen species, mostly hydrogen peroxide and hormones and electrical signaling, is regulating systemic acquired acclimation. Systemic acquired acclimation is not only associated with a systemic induction of genes, but in this paper we shows that in fact the leaves which are undergoing systemic acquired acclimation are more tolerant to the excess light stress, and the level of photoinhibition in such acclimated leaves is lower than in non-acclimated leaves. And at the beginning of 2000, we presented this model in which we shows that this redox status of the plastoquinone pool is an important regulator of the many genes, not only ascorbate peroxidases in abiotic stress responses, but also in biotic stress responses such as PR1, for example, pathogen resistance 1 gene, which is a robust molecular marker of the pathogenesis. And it is involving the others hormonal signaling like salicylate, salicylic acid, ethylene, abscisic acid, and it's regulating programmed cell death and so on. And it's depending on the glutathione metabolism and ascorbate metabolism. This was a pretty important finding, yes, and described in the other papers. Yes, in general, systemic acquired acclimation is associated also with a systemic acquired resistance. We know now, we did the experiments in 2010 when we, and 2008, published the experiments in 2 plant cell articles in which we could immunize plants only with the excess light stress. So in fact, the abiotic stress responses induce immunodefenses in plants against the virulent pathogens. So this induction was responsible for the induction of the innate immunity. And it took 15 years for the other researchers, yes, like a Dubkovsky lab, yes, just to show that the wheat strip rust resistance is depending somehow on the chloroplast retrograde signaling and ascorbate peroxidases function in the chloroplast. So during that time, in first decade of the 2000, we published several papers like evidence for direct link between the glutathione biosynthesis and stress defense gene expression in Arabidopsis. This work was pretty important because we screened over 100,000 mutants and found only 2 mutants related to the glutathione synthesis. Then chorismate synthase 1 is required for thylakoid organization, optimum plastoquinone redox status, state transition in Arabidopsis thaliana. Then we discovered that the C3 CAM transition is regulated by the redox status of the plastoquinone pool and photooxidative stress. And the most important discovery it was made that when we start to employ the lesion simulating disease mutants, and we found that LSD1 is required for acclimation to conditions that promote excess excitation energy. This was a pretty important finding, showing that the immunoresponses are somehow connected to the chloroplast retrograde signaling. And later on, we published the two plant cell articles like lysogenous arrhythmia formation in Arabidopsis is controlled by lesion simulating disease. Chloroplast signaling and lesion simulating disease 1 regulate crosstalk between light acclimation and immunity in Arabidopsis. In this last article, we showed that, and in the first article, that runaway cell death regulated by LSD1, EDS1, and PAD4 are strictly dependent on photorespiration and CO2 concentration. We could revert the runaway cell death phenotype in LSD1 mutant in a higher CO2 level, by the higher CO2 level. This was a pretty important finding. And the summary of these results are presented, so we were able to connect the redox status of the plastoquinone and signaling with the LSD1, PAD4, and EDS, and just in general regulating the cell death, acclimation, systemic acquired acclimation, systemic acquired resistance, lysogenous arrhythmia, aging, and so on. And we can see that these responses can be ranging from the nano and microseconds to the minutes, hours, and days. And later on, we discovered the electrical retrograde signaling for the cellular light memory and systemic acquired acclimation and resistance, that depending on the PQ pool, PSBS, and NPQ. In 2010, we published this article, Evidence for a Light Wavelength Specific Photoelectrophysiological Signaling and Memory of Excess Light Episodes in Arabidopsis. This article was showing in general that the electrical signaling and membrane polarization and electrical signaling which is transmitted from cell to cell, yes, is important regulator of systemic acquired acclimation and systemic acquired resistance. And in fact, we prove this systemic signaling is pretty complicated because it is set up for the regulation of the energy flow between the chlorophyll fluorescence, photochemistry, and heat. The rosette on the left side, this is the one rosette, 1 hour before and 1 hour after. And just before it is rosette from the low light when imaging is showing non-photochemical quenching. And different leaves of these rosettes, they have a different setup between the division of the energy on the between fluorescence, photochemistry, and heat. However, when you shone the excess light on the selected free leaves, then the NPQ in systemic leaves, which are still in shadow, in low light condition, are setting up one value, mostly one value. This is very important because it is showing that the conformation of the photosystems is changed, yes, between local and systemic leaves. Whenever we have a conformation and the light antenna are increasing the distance from the reaction center, then there is a more fluorescence and more heat is produced and less energy is coming for photochemistry. Whenever they are closer, yes, there is a more energy coming for photochemistry, and this conformation is changed in systemic signals. Here we can see the demonstration that the electrical signaling is regulating non-photochemical quenching. It is pretty important to know that here we didn't apply a high light for the induction of NPQ, but it was a wounding. And what wounding has to do with excess light? This is a good question. We have to see that the non-photochemical quenching are in somehow signalizing many different stresses, like a wounding, pathogen attack, excess light, and so on. It's a major sensor of the acclimation and resistance response. And what is this memory? How it was induced? When we infect the low light growing plants, low light acclimated plants with a virulent pathogen, then the pathogen within 3 days is amplifying in the leaf from 3,000 to the 10 millions. And this we can see. However, when we shone on the rosette with excess light for 1 hour, and then the plants return to the low light condition and were infected 1 hour after, 8 hours after, or 24 hours after, they got resistance towards the virulent pathogen. So virulent pathogen became avirulent pathogen because innate immunity level was induced in Arabidopsis. So in fact, systemic acquired acclimation is able to induce systemic acquired resistance. This is a very important finding. And to show a little bit more how it is complicated, the system, that this is not only physiological redox changes or hormonal changes, but also molecular quantum changes. We employ time-correlated single photon counting just to see how the NPQ is changed in local and systemic leaves. And this was published in this paper in 2015, where Kamil Czyszak and Milena Kulasek were main authors or leading authors of this finding. And in general, we tested the fluorescence decay time in exposed in systemic leaves and in leaves treated with DCMU, having a wild type and NPQ for one mutant, which is deficient in NPQ and deficient in PSBS protein, functional PSB protein. And we see that this fluorescence decay times, they behave very differently in comparison, in mutant in comparison to the wild type. In the mutant, we have a longer fluorescence decay time, yes, treated with a DCMU or not treated with a DCMU or treated with a DCMU, yes. However, when we shone on excess light for 1 hour, the fluorescence decay time in the NPQ mutant was shorter. In systemic leaves, it was also changed. In general, we can see in a table that this weighted average and arithmetic average times were different with DCMU. DCMU is giving a 2 times longer fluorescence decay time. What does it mean? That there is a less energy dissipated by the fluorescence if there is a time longer. Yes, and shorter time is a more energy dissipated by the fluorescence, this indication. And we can see the clear differences between the local systemic leaves and between the NPQ mutants and wild type and other photosystem II antenna mutants. In general, when we take the after stress memory, so-called after stress responses, so-called stress memory, then we can see that NPQ mutant cannot establish what fluorescence decay time they should have. The PSBS protein is required for the precise definition of the fluorescence decay time. The mutant is wobbling from short to long time, short to long. It is like being drunk, yes, from side to side. And later on, we published this paper in that photosystem II 22 kilodalton protein level, a prerequisite for excess light inducible memory, close tolerance to the UVC and the regulation of electrical signaling. This is a pretty important paper because it's showing that the PSBS and non-photochemical quenching has a much wider role than we thought before. It is not only responses to excess light, but it is a cross tolerance, it's a memory of stress, and it's a electrical signaling. In electrical signaling, we can clearly see that the NPQ 4 mutant wild type or overexpressor of PSBS has a very different characteristic of the electrical signaling. And recent discovery published in the Plant Cell above ground plant-to-plant electrical signaling mediates network-acquired acclimation. Yes, we ask ourselves if the electrical signaling is regulating inside in the plant, the systemic acquired acclimation and systemic acquired resistance, we ask ourselves if such electrical signaling can be transmitted from plant to plant. And bingo, yes, it could be transmitted, yes, and it is associated with changes in non-photochemical quenching, as we can see here, changing with a reactive oxygen species, changing with the electrical signaling. So this electrical signaling is coming up on the surface of the leaf, yes, and spread on the surface of the leaf and can jump from leaf to leaf if the leaves are physically connected and electrically connected. This we can see very clearly. And this we presented in a model summarizing network-acquired acclimation, and we can see that it is associated with the electrical signaling, with the ROS signaling, with the NPQ changes, with the regulation of superoxide dismutases and ROS scavenging, and of course the transcription activation of ascorbate peroxidase, ZAT12, and so on. But what is the key to this process? That NPQ changes are prerequisite for this wave of changes between the plants, and it can jump from plant to plant, from plant to plant, inducing network-acquired acclimation, systemic acquired acclimation, network-acquired acclimation, systemic acquired acclimation. So, in summary, we can summarize this presentation. We know that the photosystem 2 conformation and the energy division between the fluorescence heat and photochemistry is pretty important, and it is signalized by electrical ROS hormonal signaling to the systemic leaves. And now we know that even between the plants, the same system, yes, when they are touching each other, the electrical signaling can change the system and induce systemic acquired acclimation in the nearby plant. And in conclusion, given a few leaves per plant and hundreds of plants in contact in a meadow, thousands of cells per leaf, several dozen chloroplasts per cells, and thousands of photosystem 2 per chloroplast, which are potentially involved in the network of connection, this reveals the complexity of the systemic acquired acclimation and network acquired acclimation signaling and communication mechanism. Plants are evolutionarily smart and intelligent. They communicate in sophisticated ways, globally regulate, absorb energy flows, regulate temperature, count photons, have a quantum singlet oxygen, and physiological redox memory, acclimatize and immunize with excess absorbed energy, or with the energy absorbed in excess. Thank you very much for your attention, and I will finish with this cartoon, my presentation. Did you know that one cabbage is saying to the another, share 50% of our DNA with human? Seriously, those primitives, they can't even photosynthesize. Thank you very much.